Okay, so it's a pleasure for me to present to you all Dr. Reines. She's a biomedical scientist who studies the early events in cancer that lead to gastric cancer. Her research focuses on Helicobacter pylori, as this is a bacteria that often promotes cell transformation. She uses advanced organoid models to study gastric cancer and to see how bacteria affect the cells, the epithelial cells, and disrupt their cell behavior and promote cancer formation, as well as TP53 mutations. MAD uses advanced technologies such as CRISPR and infection models and patient-derived organoids to look at the molecular mechanisms that drive cancer initiation and to explore some new therapeutic opportunities. She carried most of her research work out in the Max Planck Institute in Berlin, establishing and optimizing these organoid models using gene editing techniques. And she's still she's just moved back to Spain. So she was in Berlin for 12 years or more. And now her research is moving towards more complex models combining tumor cells with immune cells and fibroblasts to create these more physiological relevant systems to study better the molecular mechanisms of gastric carcinogenesis. So thank you for being with us.
Thank you very much. Thank you for the kind introduction. It's a pleasure to be here today. I'm really happy to be in Spain, actually. It's my first talk back in my country. And it's super nice to see the center, met the director and everything. So as you can see here, I just put 3 institutions as affiliation because I just moved not even month ago to Madrid, and I started being a teacher in Universidad Francisco de Vitoria. But basically today what I will present was done in the Max Planck Institute and some of the revision already in the Barfield lab. So basically, to tell you a bit about my CV or how I did my scientific career when I ended my PhD, I did it in basic microbiology, like only bacteria, core bacteria. And when I finished, I had the feeling that I need this other side of the coin of this host pathogen interaction. And I moved to Berlin looking for new models for research. And I was lucky enough that in the Max Planck, in the lab of Thomas Meyer, they described as the gastric organoids. We in collaboration also with the Sina Barfield. And I started there, and they were super new. They were publishing in the boom of the organoids, because there is now, I think, a lot of tissues are already done and are ready to be organoids. But at that time, it was very at the beginning. There was no CRISPR still there, so I was lucky enough to be in the starting point there. And then I was there until 2000. Well, I got kids and everything, and then I went in 2022. Sina Barfield came to Berlin with a professorship, and it was an excellent opportunity because she was working with Hans Clevers, which is the reference in this field of organoids, and she was looking for someone to really lead the organoid platform, let's say, because she has a lot of biobanks and everything, not only from the gastric, but for the whole gastrointestinal tract. So I got also experience with that. And I went to TU Berlin, which is a technical university, and it's a biotechnology department. So what they want is to really do really advanced models and try to not only have the epithelial part, but also mix with other cell types, because at the end, we always have kind of these criticisms in organoids that we only see the epithelial response, and we don't have these other cell types that could be also important. So I was focusing the last 3 years, or 2, because also I have a break with the kids, to really improve these models and try to make an optimal conditions to then have more cell types in the system. But today, I will tell you about this story. I will try to give you some key, using organoids, we want to reveal a key mechanism on how a healthy gastric cell can then be intestinal metaplasia, which is a very important precancerous lesion in gastric cancer. So I will uncover the role of P53 in that, and I will also show you how the P53 loss has a role in defense in these cells. So before starting, I would like to, this is working, this. Yes. So I would like to introduce you a bit to the pathology. I'm not sure how is the knowledge here, but I will. So in basically gastric cancer is very diverse as all the cancers, but we in the lab focused on the intestinal type of gastric cancer. And normally it has been described to progress through a histological or pathological steps called the chorea cascade. So basically, the pathology starts with a normal mucosa that get infected with Helicobacter pylori. And this bacteria is a gram-negative bacteria that colonize approximately 40% of human stomachs. And when you get the infection, what you get in the stomach is a normal non-atrophic gastritis, which is the gastritis that we all have when we are kids. But this bacteria has developed a very sophisticated mechanisms to persist for years in the stomach. And if this happens, then what could develop the stomach is atrophy, which basically, as you can see here in the histology images, the stomach lose the gland shape and there is atrophy in the stomach. If this then persists, could be that then the stomach of the patient can develop focal intestinal metaplasia, which is focal points in which there is no gastric cells almost, and there is intestinal cells popping up in a focal way. And then at the end, what can happen is that then these patients can develop gastric cancer. As you can see in the skin, Helicobacter pylori infection is active only until atrophy stage. When the glands start to have intestinal metaplasia, the bacteria is not able to survive there, and the pathologist cannot detect Helicobacter anymore. Although it has been described that this bacteria is related to the progress towards gastric cancer and is recognized as carcinogen 1 by the World Health Organization, here they don't find it anymore. So basically, the doctors, what they can do, so the treatment can be done until here because it can be reverted, all this progress with antibiotics. However, upon intestinal metaplasia, it's a reversible process. These patients go to surveillance every 6 months. But it's irreversible because there is no involvement. They cannot be treated. Sometimes they can be treated with antibiotics, but doesn't mean that it has to be efficient because the bacteria is not there anymore. So basically, this is the point of no return of this progression. What is this? So intestinal metaplasia, well, metaplasia in general, is the presence of a cell that should not be there in that tissue. So it's a shift from one type to another type. And in the case of intestinal metaplasia in the stomach, it means that the gastric cell starts to be an intestinal cell. So taking into account this, I wanted to define a bit more. So how can we define intestinal metaplasia? So basically, histologically and pathologically, there is several of differentiation markers and transcriptional factors that are described to be changing in the cell. So basically, a normal cell, a gastric cell, has gastric markers like the mucin MUC5AC or MUC6, that are mucins very important, for example, for infection with Helicobacter, and also has SOX2, which is a very important marker in development. However, if something happens, then there is a switch. So there is the gastric markers that starts to be downregulated and new markers appear. Like, for example, this mucin 2, which is the mucin of the intestine, and pylin, for example, which is also expressing the enterocytes of the intestine, and another transcriptional factors very important in the intestinal development is CDX2 and CDX1. So basically, with this, the questions was like, if this is really important for the progression to the disease, we had two questions. How does intestinal metaplasia develop in the stomach? So we know the markers that are switching, but what is the trigger that makes this cell suddenly become an intestinal cell? And what makes this process irreversible? Because if we think about infection, we can revert it with antibiotics, but what is it that it stays and then the patient cannot be treated anymore? This was one of the first questions. And the second question, why Helicobacter pylori is unable to survive here, why we don't find it anymore, and it's able to persist all over, but then when the glands undergo intestinal metaplasia, then there is no bacteria anymore. So to do this, we thought, okay, to do a process irreversible, we have to do a mutation, because otherwise it can be that the infection epigenetic can be also reverted. So we went to the literature back in 2015. And there was several studies that they described the mutational landscape of gastric cancer. So it's not like the colon cancer that it's really clear that one mutation happened after the other until you have the tumor. But all over, both studies characterize TP53 as the more mutated gene in gastric cancer. So we said, okay, it's 50% of the whole thing, but let's try with it and see if we can see something there. Also, not only in gastric cancer, there was also some studies that P53 was already mutated before having cancer. So it was a good candidate to see if with mutating P53 we could get a hit in where intestinal metaplasia could rise. So to do this, we, as I said in the beginning, I was lucky enough to be in a lab that they were having the gastric organoids, and we used this model. I guess you have heard about organoids several times, but I will briefly explain how we get the organoids from, we got the organoids from Reines. So basically, they are adult stem cell derived organoids that are not pluripotent. They come exactly from the tissue where we want to then represent the organoids from. And basically, we have a sleeve resection from a metabolic surgery from obese people. We get a piece of stomach and we extract the epithelial cells. Nowadays, we also extract the fibroblasts, and we can try to get some migrating lymphocyte there. So we get the stem cell from there and we put it in matrigel, which is the extracellular matrix. And we do a combination of growth factors and morphogens that after 10 days, we get this beautiful organoids, which is structured, polarized, so we can get, let's say here inside the organoid will be the lumen of the stomach, and outside the organoid will be, let's say, the basal side of the cell, of the epithelial cell. So here, basically, will come the fibroblast and all other cells that migrate into the organoid. So, and then these cultures can be forever grown, let's say. At some point they die, of course, but for example, we can do biobanks out of it. So the first isolation, we can freeze them down and we can have them forever if we want to then defreeze them again. Not only now, we use it from healthy individuals, but we also have a biobank of tumor organoids from the whole gastrointestinal tract that this also can be used to try several drugs. Well, so we have this model. So I use this gastric healthy organoids and TP53 mutation, and I use CRISPR-Cas9 technology to mutate the gene specifically. So basically, I explained here how we did it. So it's the setup of the experiment. So we have these wild-type human gastric organoids that came from 3 different patients. This is also very important. We get the significance when we try how the mutation affects in different patients, not only in one. So we design guide RNAs against P53, and we transduce it to the single cells. So we basically break the organoids into single cells, and we transduce the lentivirus that bears the guide RNA against the target gene that we want to mutate, and also the Cas9 that is bound to GFP. So with this technique, we get green organoids that we are sure that express Cas9. So at least the protein that should cut the DNA is inside the organoids. And then P53 is a nice target because if you mutate the transporter or something, you have to be sure that you have to sequence to see that the gene is not there anymore. However, if you mutate P53, there is a functional state that you can select the mutants. And this is with the treatment with Nutlin. Nutlin is a drug that targets MDM2, which is let's say, the protein that degrades P53. So if P53 is there and you treat with Nutlin and you remove the MDM2 with the drug, then the wild type cells will just die because P53 will be all the time active and will bring the cell to apoptosis. However, if I mutate P53, the cell will not have P53 and the mutants will survive. So this was interesting to just select exactly the mutants of P53. Then I did clonal expansion to have a culture that comes only from one single cell, and I did it on feeder cells. Basically, it's kind of a fibroblast line that you put the epithelial cells on, and then you really see individual colonies of epithelial cells growing, and you can pick them and grow them again back in matrigel, and then you get several clones again on matrigel that grew as a single clones mutated with P53. So we had several, as I said, from 3 different patients, and this is amplicon sequencing to check really. We were kind of sure because, as I said, this functional selection is really efficient, but of course we have to demonstrate which kind of mutation because CRISPR can be that it has deletions bigger or smaller or so on, and we have to be sure also if both alleles were mutated of the organoids. So here this table, maybe it's a bit confusing, but I just wanted to focus, for example, in this clone. So here is the two alleles, and D means deletion. So one allele has a deletion of 33, and the other allele has a deletion of 4 nucleotides. So here is the proportion of the alleles. This is the total shift, and as you can imagine, since 33, this 33 has an in-frame mutation. So, and here the other allele has a frame shift. And the others, for example, most of them had the friendship, so P53 was completely abolished. However, if you see then the Western blot, in this case that I just mentioned, that one that has an in-frame mutation in one allele, P53 was still there expressed. But although not functional, but still we had a bit of expression of P53. So it was basically very important to characterize how were these cells, which alleles were mutated, and how P53 was expressed there. And actually, I know that this, for example, in this case, this clone is not functional P53 because we went to RNA-seq. We bring them all to RNA sequencing to see what was the effect of not having P53 in there. And we saw that all target genes of P53, which is these Fisher target genes, they were all downregulated in all the mutants. So having this, and as I said, I bring them to RNA-seq. We saw, okay, what are the effects of not having P53? When we compare all of them, one of the first thing that popped up was the differential stomach and intestinal markers. Here I put it a bit bigger. This is with the 3 patients, the average of them. So basically all the gastric cancer, all the gastric markers were downregulated. As I said before in my introduction, these cells that didn't have P53, didn't have MUC5AC, didn't have MUC6, but started to have more and more intestinal markers. And I label in red the CDX2 because it's the expression of CDX2 is a very important transcriptional factor in intestinal development, and actually it's the master regulator. So CDX2 is up. All the other genes, let's say MUC2, the mucins, it's regulated by CDX2, so it was very important to see this up. And also this DMBT1, which is a new target that I will explain you later. Also, we did gene set enrichment analysis, and we saw that suddenly all the clones had a specific enrichment for intestinal specific markers, and not only intestine markers, but also then we use a gene set of intestinal metaplasia, and all of them showed a clear enrichment for intestinal metaplasia gene sets. So we validate all of this by qPCR, we check for CDX2 expression, so indeed they hide and not only express, but also translocated into the nucleus, where this transcriptional factor has the function. So what it means that all these markers are changed, what it means at the cellular level. So we went in and we went to the microscope and we saw that the P53 knockout cells started to have goblet cells, which is also a very, very characteristic cell present in intestinal metaplasia when the pathologists go into the stomach of the patient. So, and we had several ones, and then also we checked for goblet cells in another technique, which is this staining. And here you can see the goblet cells. This is the wild type organoid that doesn't have any, but then here we have our mutants, which are full of goblet cells. Full, but like here, for example, 3 or 4 cells per organoids. Saying that maybe P53 loss function leads to an intestinal differentiation of gastric organoids toward intestinal metaplasia. So P53 is described as a tumor suppressed as a guardian of the genome, but here we describe a function of P53 determining cell fate when it's gone. So as our first question that we had, we could just say that maybe one of the key mechanisms that lead to the switch of a gastric cell to the intestinal cell is the loss of a very early stage of P53. The next question was, why then we don't have a Helicobacter here in this stage? So now with this model that we have with the organoids, we suddenly had a model, an in vitro model to study intestinal metaplasia. So next what we did, we infect the intestinal patient model with Helicobacter pylori. And voila, the addition of the bacteria was decreased in the clones. And actually not only this, visually, the wild type cells were really infected, but the mutant organoid, this is an infection in 2D because also the organoids I didn't explain, but you can also smash them, put it into single cells and culture them and differentiate them and have any type of cell of the stomach that you like, just changing the media. So what I did is to put them in 2D and infect them. I could also microinject, but it's more tedious, so I just decided to do it in 2D. And as you can see, the knockout organoids or the metaplastic organoids cannot be infected, and this is quantified here by the hummingbird phenotype, which is also another readout very common in Helicobacter pylori field. So we went back to our RNA-seq, and we suddenly saw that there is a lot of antimicrobials upregulated. DMBT1, the one that I have said at the beginning, 50-fold upregulated. So we went to the literature and see what is DMBT1, because I never heard about it. And actually, there was not much about it, but it was a glycoprotein with an SRCRP2 bacterial binding, which was very promising, and it's a pattern recognition molecule. And there were some papers very old that they did some binding experiments with other bacteria like Salmonella, Streptococcus, or Pseudomonas. And actually, if you see, the name is deleted in malignant brain tumors. So it was like, what is doing a brain tumor deleted thing in the stomach? But well, actually, it was just the name because it was discovered there. But then all the functionality of DMBT1 was done in infection, actually. Then I checked all the publications, and they were in some microarrays in the monkeys. DMBT1 was upregulated in a CagA-dependent manner. CagA is the main virulence factor of Helicobacter pylori. So this was just, I'm just throwing here some data of the reference that I found. But also another paper showed me that DMBT1 was also one of the top 25 genes upregulated in intestinal metaplasia. So kind of all the literature was pointing out that was a good candidate to see why Helicobacter is not there. So here is a staining of that paper where it shows that DMBT1 is upregulated there. And as you can see, it's very much expressed in the intestinal metaplasia and not in the normal. So, and also I checked protein atlas, and it was basically expressed in all the gastrointestinal tract. So I went to my organoids and I checked and I validated. So my organoids express very much DMBT1, and also it appears as a granulated protein inside the cell. So here is a staining of one of the clones with DMBT1. So next, and logically, I did a lot of functional assays to see if DMBT1 as the recombinant protein was interacted with the Helicobacter pylori. I first checked the images, so I went to infect my knockout organoids, and I see that the P53 knockout organoids were secreting DMBT1 in red, and DMBT1 was completely surrounding the bacteria. And then I did some functional assays to check this binding, and eventually I demonstrate that H. pylori, also DMBT1, the recombinant protein, really binds to H. pylori and not to other bacteria, for example. And the consequence of this binding of DMBT1 is the agglutination of the bacteria. I did, again, functional assays to determine this, and I see that it's depending on the concentration of DMBT1 agglutinates H. pylori. So then I sit back and say, okay, what does it mean that an epithelial cell that is mutated with P53 secretes an antimicrobial, DMBT1? What could it be, the implication in vivo? What would happen that just a cell that is suddenly mutated start to have some antimicrobials? What could happen? So my idea was that this secretion, well, before going to here, I want to show you more pictures. You can see also here in the stainings that DMBT1 really agglutinates the bacteria, and actually where the bacteria is, it looks like there is even more intensity of DMBT1 secreted, like in an active way. So as I said, what is the meaning of having these bacteria in one side in the apical site of the infection, having bacteria agglutinated? So it was not killing, because if I just have 50% of reduction of survival of Helicobacter, but something was doing, because the bacteria was not able to move. It was completely agglutinated. So I exposed the bacteria and DMBT1 to macrophages and neutrophils to see if in the presence of DMBT1, these immune cells will be more active to eat it. In the idea of course, in my much more complex models, like that I will have both type cells, the epithelial and the immune cells, I would eventually see a migration of immune cells into the epithelial cells to really kill the bacteria. That is my dream experiment. So what I did is to expose to infect the macrophages with the Helicobacter pylori, either with or without DMBT1. And as you can see, there was much more phagocytosis of Helicobacter when DMBT1 was there. And the same with the neutrophils. I saw more phagocytosis if DMBT1 was there, or the bacteria was pre-incubated with DMBT1. So, and then of course it came the questions of the reviewers, and they were saying, what happened with vivo? So we got the DMBT1 knockout, and indeed we demonstrate that DMBT1 it's a good antimicrobial, a new antimicrobial against Helicobacter pylori, because when we infect the DMPT1 knockout mice, we saw much more colonization of the mice. So we can say that loss of P53 triggers epithelial defense against H. pylori via DMPT1 secretion. Then again, the question that we got, what happened in vivo? So we get also the P53 knockout mice, and we saw that the DMPT1 was upregulated in the knockout. And then when we infect them, we also see here, it's not very visible, but you can see that there is red spots where Helicobacter is in red. And when there is DMPT1, Helicobacter is not present. This is quantified also here. And then we are back to our questions, and we can say that maybe Helicobacter cannot colonize because there is an active epithelial response in the intestinal metaplasia. Being DMPT1 one of the main antimicrobials, actually new, because nobody still had described the interaction with Helicobacter pylori. And we described as a new antimicrobial against Helicobacter in intestinal metaplasia. Now, as a translational institute that we are, what's going on in the patients? So we collaborate with the pathology department in Berlin, and we checked our targets in the patients. So we saw that here I present a gastritis sample, which is an active infection of Helicobacter pylori. And here I show the cut of intestinal metaplasia of patients. So, and here is the staining of MUC2 that I said at the beginning, that MUC2 is a mucin that is just present in the intestinal metaplasia. Here is an active infection, and here is not there, sorry, MUC2, because it's gastritis, still didn't transform. And here in intestinal metaplasia, we have glands that already have focal expression of MUC2. When I checked for DMPT1, same happens. So healthy epithelium doesn't have DMPT1, but intestinal metaplasia has the expression of DMPT1 in the epithelium. And then when I check Helicobacter pylori is only active in the gastritis sample when there is an active infection. But when we reach the point of intestinal metaplasia, it's not there anymore. Also, another of my dream experiments or my dream project would be to have the same patient with this coming from the gastritis sample, an atrophy sample, if it's possible, intestinal metaplasia, and if I hope not a gastric cancer, but all the cardiac cascade from the same patient and analyze this and this transition all over all the cascades. And this I demonstrated with 25 different patients in the same manner. So this is more or less it. So here I present the highlights of this paper that hopefully will be seen the light very, very soon. So we said that we described that the loss of P53 in gastric organoids leads to differentiation of the gastric cells towards intestinal cells, suggesting a role of P53 loss in promoting intestinal metaplasia in early stage. So the loss of this P53 confers protection against H. pylori infection, mainly because of DMPT1 secretion. DMPT1 as functional assays binds and agglutinates H. pylori, being a novel antimicrobial agent for this pathogen. In vivo, P53 loss of function induced DMPT1 upregulation, and in consequence, we observe H. pylori colonization. We observe less H. pylori colonization in the antrum glands. And in human tissue, metaplastic glands upregulate DMPT1 and show loss of active H. pylori infection. So with this, I would like to conclude, and of course, acknowledge Bartel Lab, because I got a lot of training for the last years to improve these models, because we, as I said, my dream experiment would be to see all of this in a much complex model. And yeah, with a lot of cool science, but still I cannot talk about it. And then I also would like to say thank you to the Laboratory of Infection Oncology, which is my former boss that was in the Max Planck, moved to Kiel in the north of Germany and built a lab there. And I'm kind of in the middle of publishing this data, which is from this lab. So I'm still in contact with them, and I'm supervising 2 students there to just continue with the work, actually finish it, because they're almost done. Also the hospital, where I have 2 amazing medical doctors that help me also with this, all the pathology department of the Charité, and then Other departments from the Max Planck that I'm still in contact, the microscopy core facility and the Department of Cellular Microbiology that allows me to have the immune cells for then these complex models. And then the University of Dundee, because we have another project in parallel where because P53 loss, we mutated, but then there is a lot of isoforms, that it's a new complete word. There's conference just about isoforms. And I'm in contact with this Jean Bordon because he's a very expert on P53, and he would like to see also the involvement of this new function in infection. And with this, then the function. So with this, I would like to thank you all for listening, and I'm open to the questions.